Microscopic techniques. ChatGPT said. Microscopy is an indispensable tool in zoological research, enabling scientists to observe and analyze structures that are invisible to the naked eye. The evolution of microscopic techniques has significantly advanced our understanding of animal morphology, physiology, and developmental biology. This comprehensive overview delves into various microscopy methods, their principles, applications, and relevance in zoological studies. 1. Optical microscopy. Optical microscopy, also known as light microscopy, utilizes visible light to magnify specimens. It encompasses several techniques. Bright field microscopy, the most conventional form, where light passes directly through the specimen. It's suitable for observing stained or naturally pigmented samples but offers limited contrast for transparent specimens. Dark field microscopy employs oblique illumination, causing light to scatter and making the specimen appear bright against a dark background. This technique enhances contrast in unstained, transparent samples, revealing fine structural details. Thick contrast microscopy, introduced by Fritz Cernick, this method converts phase shifts in light passing through transparent specimens into variations in intensity. It's particularly useful for examining live cells and tissues without staining. Appearance contrast Dick microscopy, also known as no-Marcy microscopy, Dick uses polarized light to produce high-contrast images with a pseudo-3D effect, aiding in the visualization of fine structural details in live specimens. Fluorescence microscopy relies on the emission of light by fluorophores within the specimen when excited by specific wavelengths. The technique is invaluable for localizing specific proteins, nucleic acids, and other molecules, often using fluorescent dyes or genetically encoded markers. 2. Chemical laser scanning microscopy (CLSM). CLSM enhances optical microscopy by using laser light to scan specimens point by point, constructing high-resolution images with improved contrast. It allows for optical sectioning, enabling the reconstruction of three-dimensional structures, which is crucial for studying complex tissues and embryonic development in zoology. 3. Electron microscopy. Electron microscopy offers magnifications up to 2 million times, surpassing the limits of light microscopy. It includes tension electron microscopy. TEM transmits electrons through ultra-thin specimens, revealing internal structures at the molecular level. TEM is essential for examining cell organelles, viruses, and macromolecular complexes. Scanning electron microscopy (SEM) scans the specimen's surface with a focused electron beam, producing detailed three-dimensional images. SEM is used to study surface morphology and topography of specimens, such as insect exoskeletons and tissue surfaces. 4. Scanning probe microscopy. The category includes techniques like atomic force microscopy (AFM), which measures forces between a sharp probe and the specimen surface to generate topographical maps at the atom level. AFM is in studying the mechanical properties of biological specimens, including cell membranes and biomolecules. 5. Sample preparation techniques. Proper specimen preparation is vital for accurate microscopy. Common methods include fixation, preserves tissue morphology and molecular integrity, typically using chemical agents like formaldehyde. Embedding, involves infiltrating tissues with media such as paraffin or resins to provide support during sectioning. The paraffin method is widely used for preparing thin sections for light microscopy. Alternatively, the cell-laden method is employed for larger, harder tissues, while the freezing method is used for rapid sectioning without chemical fixation. N. Wikipedia.org. Sectioning, slicing embedded tissues into thin sections using microdomes or ultramicrodomes, essential for TEM analysis. Staining, applying dyes that bind to specific cellular components, enhancing contrast and enabling differentiation of structures. Instance, hematoxylin and eosin H&E. Staining is commonly used to distinguish between nuclear and cytoplasmic components. 6. Advanced techniques. Modern microscopy has evolved to include sophisticated methods. Live cell imaging utilizes time-lapse microscopy to observe dynamic processes in living cells, such as cell division, motility, and intracellular transport. Super-resolution microscopy breaks the diffraction limit of light, achieving nanometer-scale resolution. Techniques like stimulated emission depletion (STED) and photoactivated localization microscopy (POM) fall into this category, allowing visualization of molecular interactions at the nanoscale. Correlative light and electron microscopy (CLEM) combines the advantages of light and electron microscopy to correlate dynamic live cell imaging with high-resolution structural information. 7. Applications in zoology. Microscopy techniques are pivotal in various zoological research areas. Taxonomy and systematics, detailed morphological studies aid in species identification and classification. Developmental biology, observing embryonic development stages provides insights into growth patterns and congenital anomalies. Histology and cytology, examining tissues and cells elucidates normal physiology and pathological conditions. Neurobiology, investigating nervous system structures and functions enhances understanding of neural networks and behavior. Parasitology, identifying and studying parasites at the microscopic level informs disease control and prevention strategies. 8. Solved previous year question. Question. Describe the principle and applications of fluorescence microscopy in zoological research. Answer. Fluorescence microscopy operates on the principle that certain substances emit light upon excitation by specific wavelengths. In this technique, specimens are labeled with fluorophores, which absorb light at one wavelength excitation. Sources. You said. Visualization of cells and subcellular components by light microscopy. ChatGPT said. Visualizing cells and their subcellular components through light microscopy is fundamental in zoological research, providing insights into cellular architecture and function. This comprehensive overview explores various light microscopy techniques, their principles, and applications in studying cells and subcellular structures. 1. Bright field microscopy. Bright field microscopy is the most conventional form of light microscopy, where light passes directly through the specimen. This technique is suitable for observing stained or naturally pigmented samples, offering clear images of cell morphology and tissue architecture. However, it provides limited contrast for transparent specimens, necessitating staining procedures to enhance visibility. 2. Phase contrast microscopy. Developed by Fritz Zernick, phase contrast microscopy converts phase shifts in light passing through transparent specimens into variations in intensity. This method is particularly advantageous for examining live, unstained cells, allowing researchers to observe dynamic processes such as cell division and motility without altering the specimen. 3. Differential interference contrast DIC microscopy. Also known as Nomarski microscopy, DIC employs polarized light to produce high-contrast images with a pseudo-three-dimensional effect. This technique enhances the visualization of fine structural details in live specimens, such as organelles and cytoskeletal elements, by detecting differences in refractive indices within the cell. 4. Fluorescence microscopy. Fluorescence microscopy relies on the emission of light by fluorophores within the specimen when excited by specific wavelengths. DIC is invaluable for localizing specific proteins, nucleic acids, and other molecules within cells. By using fluorescent dyes or genetically encoded markers like green fluorescent protein, GFP, researchers can study the distribution and dynamics of subcellular components with high specificity and sensitivity. 5. Confocal laser scanning microscopy, CLSM. CLSM enhances optical microscopy by using laser light to scan specimens point by point, constructing high-resolution images with improved contrast. It allows for optical sectioning, enabling the reconstruction of three-dimensional structures, which is crucial for studying complex tissues and cellular interactions in zoology. 6. Sample preparation techniques. Proper specimen preparation is vital for accurate visualization under light microscopy. Common methods include fixation, preserves tissue morphology and molecular integrity, typically using chemical agents like formaldehyde. Embedding, infiltrating tissues with media such as paraffin or resins to provide support during sectioning. Sectioning, slicing embedded tissues into thin sections using microtones, essential for observing internal structures. Staining, applying dyes that bind to specific cellular components, enhancing contrast and enabling differentiation of structures. For instance, hematoxylin and eosin H&E, staining is commonly used to distinguish between nuclear and cytoplasmic components. 7. Advanced techniques. Modern light microscopy has evolved to include sophisticated methods. Live cell imaging utilizes time-lapse microscopy to observe dynamic processes in living cells, such as intracellular transport and signal transduction, providing real-time insights into cellular function. Pseudo-resolution microscopy, techniques like stimulated emission depletion (STED) and photoactivated localization microscopy (POMP) surpass the diffraction limit of light, achieving nanometer scale resolution. These methods allow visualization of molecular interactions at the nanoscale, offering unprecedented detail of subcellular structures. Lattice light microscopy. This technique uses a thin sheet of light to illuminate specimens, reducing phototoxicity and enabling high-speed, high-resolution imaging of live cells in 3 dimensions. It is particularly useful for observing rapid cellular processes with minimal damage to the specimen. 8. Applications in zoology. Light microscopy techniques are pivotal in various zoological research areas. Developmental biology. Observing embryonic development stages provides insights into growth patterns and congenital anomalies. Histology and cytology. Examining tissues and cells elucidates normal physiology and pathological conditions. Neurobiology. Investigating nervous system structures and functions enhances understanding of neural networks and behavior. Parasitology. Identifying and studying parasites at the microscopic level informs disease control and prevention strategies. 9. Solved previous year question. Question. Explain the principle and applications of phase contrast microscopy in visualizing live cells. Answer. Phase contrast microscopy operates on the principle of converting phase shifts in light passing through a transparent specimen into variations in intensity, producing high contrast images without the need for staining. This is achieved by a phase plate that alters the phase of light passing through different parts of the specimen, enhancing contrast. It is particularly useful for observing live cells, allowing researchers to study dynamic processes such as cell division, motility, and intracellular transport in their natural state without chemical fixation or staining. 10. Multiple choice questions. Which microscopy technique utilizes polarized light to produce high-contrast images with a pseudo-three-dimensional effect? A. Bright field microscopy. B. Phase contrast microscopy. C. Differential interference contrast DIC microscopy. D. Fluorescence microscopy. Answer. C. Differential interference contrast DIC microscopy. Which of the following is a significant advantage of fluorescence microscopy in cell visualization? A. Ability to observe live cells without staining. B. Enhanced contrast for transparent specimens. C. Specific localization of proteins and nucleic acids within cells. D. Visualization of surface morphology in 3 dimensions. Answer. C. Specific localization of proteins and nuclei. Sources. You said. Resolving powers of different microscopes, ChatGPT said. The resolving power of a microscope is its ability to distinguish between 2 closely spaced points as separate entities, thereby revealing fine structural details. This capability is crucial in zoological research, where understanding cellular and subcellular structures is essential. Different types of microscopes offer varying resolving powers, primarily influenced by the wavelength of the illumination source and the numerical aperture of the objective lens. 1. Optical light microscopes. Optical microscopes utilize visible light to illuminate specimens. The theoretical resolution limit of these microscopes is approximately 0.2 micrometers, 200 nanometers, constrained by the diffraction limit of light. This means that 2 points closer than 200 nanometers cannot be resolved as distinct entities. Advanced techniques, such as confocal laser scanning microscopy, can enhance image contrast and clarity but remain subject to this diffraction limit. MICROSCOPYU, com. 2. Electron microscopes. Electron microscopes employ electron beams with much shorter wavelengths than visible light, enabling significantly higher resolving powers. There are 2 primary types. Transmission electron microscopes TEMs can achieve resolutions up to 0.05 nanometers, 50 picometers, allowing visualization of individual atoms. N. Wikipedia, org. Scanning electron microscopes SEMs provide detailed three-dimensional images of specimen surfaces with resolutions around 0.4 nanometers, 400 picometers. N. Wikipedia, org. 3. Scanning probe microscopes. Scanning probe microscopes, including atomic force microscopes AFM, can achieve atomic-level resolution, enabling visualization of individual atoms and molecular structures. These microscopes are instrumental in nanotechnology and materials science research. 4. Factors influencing resolving power. The resolving power of a microscope is influenced by several factors. Wavelength of illumination. Shorter wavelengths yield higher resolution. For instance, ultraviolet light offers better resolution than visible light in optical microscopy. MICROSCOPYU, com. Numerical aperture, NA, a higher NA indicates a greater ability to gather light and resolve fine details. It depends on the refractive index of the medium between the lens and the specimen and the half angle of the maximum cone of light that can enter the lens. Lens quality and aberrations. Imperfections in lens design or manufacturing can reduce resolving power by introducing aberrations that distort the image. 5. Applications in zoology. Understanding the resolving power of various microscopes allows zoologists to select appropriate instruments for their research needs. Light microscopy, suitable for observing live cells, tissues, and organisms, especially when combined with staining techniques to enhance contrast. Ultron microscopy, ideal for detailed examination of ultrastructural components, such as organelles, viruses, and macromolecular complexes, providing insights into cellular architecture and function. Scanning probe microscopy, utilized for studying surface topography at the molecular and atomic levels, contributing to understanding of molecular interactions and structural biology. 6. Solved previous year question. Question. Compare and contrast the resolving powers of light microscopes and electron microscopes, and discuss their implications in biological research. Answer. Light microscopes have a resolving power limited to approximately 200 nanometers due to the diffraction limit of visible light, making them suitable for observing cells and larger organelles. In contrast, electron microscopes, utilizing electron beams with shorter wavelengths, achieve much higher resolutions, TEMs up to 0.05 nanometers and SEMs around 0.4 nanometers, allowing visualization of subcellular structures and macromolecules. The higher resolving power of electron microscopes enables detailed structural analysis at the molecular and atomic levels, which is crucial for understanding complex biological processes. 7. Multiple choice questions. Which factor primarily limits the resolving power of a light microscope? A. Numerical aperture of the objective lens. B. Wavelength of the illumination source. C. Quality of the specimen preparation. D. Magnification power of the eyepiece. Answer. B. Wavelength of the illumination source. What the approximate resolving power of a transmission electron microscope TEM? A. 200 nanometers. B. 0.4 nanometers. C. 0.05 nanometers. D. 2 micrometers. Answer. C. 0.05 nanometers. Which type of microscope is most suitable for observing the surface topology of a specimen at atomic resolution? A. Light microscope. B. Trenchant electron microscope. C. Scanning electron microscope. D. Scanning probe microscope. Answer. D. Scanning probe microscope. Understanding the resolve powers of different microscopes is essential for selecting the appropriate imaging technique in zoological research, ensuring that the chosen method aligns with the specific structural details under investigation. Sources. You said. Microscopy of living cells, ChatGPT said. Visualizing living cells through microscopy is pivotal in zoological research, enabling the observation of dynamic biological processes in real time. This comprehensive overview delves into various microscopy techniques tailored for live cell imaging, their principles, applications, and considerations for optimal use. 1. Wide field microscopy. Wide field microscopy is a fundamental technique where the entire specimen is illuminated uniformly, allowing for the observation of live cells with minimal phototoxicity. This method is advantageous for capturing rapid events and is often employed in conjunction with fluorescence imaging to study specific cellular components. However, out-of-focus light can reduce image clarity, making it less suitable for thick specimens. Leica Microsystems, com. 2. Phase contrast microscopy. Phase contrast microscopy enhances the contrast of transparent specimens without the need for staining, making it ideal for observing live cells. It converts phase shifts in light passing through the specimen into intensity variations, rendering detailed images of cellular structures and processes. This technique is particularly useful for monitoring cell division, motility, and morphological changes. N. Wikipedia, org. 3. Differential interference contrast DIC microscopy. Microscopy employs polarized light to produce high-contrast images with a pseudo-three-dimensional appearance. It is beneficial for visualizing fine details in live, unstained cells, such as organelles and cytoskeletal elements. DIC is especially useful in studying cell morphology and intracellular structures with enhanced depth perception. The Scientist, com. 4. Fluorescence microscopy. Fluorescence microscopy involves labeling specific cellular components with fluorescent probes, allowing for the visualization of targeted structures within live cells. While this technique provides high specificity, it requires careful consideration of phototoxicity and photobleaching, which can affect cell viability and fluorescence intensity over time. Optimizing excitation light intensity and exposure time is crucial to minimize these effects. Leica Microsystems, com. 5. Confocal laser scanning microscopy, CLSM. CLSM enhances fluorescence microscopy by using laser light to scan the specimen point by point, reducing out-of-focus light and improving image resolution. This technique enables optical sectioning, allowing for the reconstruction of three-dimensional structures in live cells. However, the increased light exposure can lead to phototoxicity, necessitating a balance between image quality and cell health. Zeiss Campus, Magnet, FSU, Edu. 6. Spinning disc confocal microscopy. Spinning disc confocal microscopy utilizes a disc with multiple pinholes to scan the specimen rapidly, enabling high-speed imaging with reduced phototoxicity compared to traditional CLSM. This method is suitable for observing fast cellular processes in real time, such as vesicle trafficking and cytoskeletal dynamic. Abidi, com. 7. Total internal reflection fluorescence TIRF microscopy. TIRF microscopy selectively illuminates and excites fluorophores in a thin region near the cell-substrate interface, typically within 100 to 200 nanometers of the cover slip. This technique is ideal for studying events that are near the plasma membrane, such as receptor-ligand interactions and exocytosis, with high signal-to-noise ratio. ANATOMYPUBS. Online library. Wiley, com. 8. Multiphoton microscopy. Multiphoton microscopy employs longer wavelength light to excite fluorophores through the simultaneous absorption of 2 or more photons. This approach allows for deeper tissue penetration with reduced phototoxicity, making it suitable for imaging live cells within thick specimens or whole organisms. It is particularly advantageous for intravital imaging and studying cellular interactions in their native environment. Abidi, com. 9. Light sheet fluorescence microscopy. Light microscopy illuminates the specimen with a thin sheet of light, reducing photo damage and enabling rapid, high-resolution imaging of live cells in 3 dimensions. This technique is beneficial for observing developmental processes and large-scale cellular dynamics over extended periods. Abidi, com. Considerations for live cell imaging. When selecting a microscopy technique for live cell imaging, several factors must be considered. Phototoxicity. Excessive light exposure can damage cells and alter their behavior. Employing techniques that minimize light intensity and exposure duration is essential to preserve cell viability. Temporal resolution. Capturing fast biological events requires imaging methods with high temporal resolution to accurately record dynamic processes. Spatial resolution. The level of detail needed dictates the choice of microscopy, with some techniques offering higher spatial resolution to visualize subcellular structures. Fluoroprotection. Choosing appropriate fluorescent probes with minimal photobleaching and phototoxicity is crucial for prolonged imaging sessions. Environmental control. Maintaining optimal conditions e.g., temperature, pH, humidity during imaging is vital to ensure physiological relevance and cell health. 11. Applications in zoology. Live cell imaging techniques are indispensable in various zoological research areas. Developmental biology. Monitoring embryonic development and cell differentiation in real time provides insights into growth patterns and morphogenesis. Neurobiology. Observing neuronal activity and synaptic interactions enhances understanding of nervous system function and behavior. Cellular physiology. Investigating intracellular processes, such as signal transduction and organelle dynamics, elucidates mechanisms underlying cell function and adaptation. Pathology. Studying pathogen-host interactions and disease progression at the cellular level aids in identifying therapeutic targets and strategies. 12. Solved previous year question. Question. Discuss the advantages and limitations of total internal reflection fluorescence TIRF microscopy in studying membrane-associated processes in live cells. TIRF microscopy offers several advantages for studying membrane-associated. Sources. Said, phase contrast. ChatGPT said, phase contrast microscopy. Phase contrast microscopy is a powerful technique for visualizing live, unstained cells. It enhances the contrast of transparent specimens by converting phase differences in light passing through a sample into intensity variations. The method is widely used in zoology for studying cell morphology, motility, and internal structures without the need for staining or fixation, which can alter cell physiology. 1. Principle of phase contrast microscopy. Light waves passing through a specimen experience phase shifts due to differences in refractive indices between cellular components and the surrounding medium. These phase differences are typically invisible to the human eye. Phase contrast microscopy transforms them into differences in brightness, making transparent structures distinguishable. A phase annulus in the condenser focuses hollow cone-shaped light onto the specimen. Light passing through the specimen is either undisturbed background light or delayed by different refractive index regions, diffracted light. A phase plate in the objective lens selectively retards or advances the phase of diffracted light. Interference between background and diffracted light enhances contrast, creating a bright or dark image depending on phase shifts. This method effectively highlights internal structures like nuclei, mitochondria, and vacuoles, making it ideal for observing living cells in their natural state. 2. Advantages of phase contrast microscopy. Visualization of unstained specimens. Unlike bright field microscopy, phase contrast allows detailed observation without staining, which is useful for studying living cells without altering their physiology. Enhanced contrast. Cellular components such as organelles, cytoplasmic granules, and membranes become visible due to increased contrast. Non-invasive technique. Cells remain viable, making phase contrast microscopy ideal for time-lapse studies in live cell imaging. Easy to use. No complex sample preparation is required, making it a quick and effective technique for biological research. 3. Limitations of phase contrast microscopy. Halo artifacts. A common issue where bright halos appear around specimens, reducing image clarity. Not suitable for thick specimens. Phase contrast works best with thin, transparent samples. Thick specimens can create overlapping phase shifts, leading to poor image quality. Limited color information. Since phase contrast relies on differences in refractive index rather than staining, it does not provide natural color contrast. 4. Applications in zoology. Cell culture studies. Observing live cell behavior, growth, and division in tissue culture. Microbial and protozoan research. Studying motility and morphology of bacteria, protozoa, and microscopic aquatic organisms. Sperm motility analysis. Assessing sperm viability and movement in reproductive biology. Neuroscience research. Observing live neurons and glial cells to study cellular interactions in real time. Parasitology. Examining live parasites and host parasite interactions in unstained preparations. 5. Solved previous year question. Question. Explain the principle of phase contrast microscopy and discuss its advantages and limitations in studying live cells. Answer. Phase contrast microscopy exploits differences in the refractive indices of cellular structures to generate contrast. It uses a phase annulus in the condenser to direct light as a hollow cone onto the specimen. Light that passes through cellular components undergoes phase shifts, which are selectively altered by a phase plate in the objective lens. Interference between background and diffracted light enhances contrast, allowing visualization of transparent, unstained structures. Advantages include non-invasive imaging of live cells without staining, enhanced contrast for internal cellular components, simple and rapid observation of biological samples. Limitations include the presence of halo artifacts that may distort image clarity, ineffectiveness for thick specimens due to overlapping phase shifts, lack of color contrast, requiring alternative methods for specific molecular identification. Overall, phase contrast microscopy is a valuable tool in zoological research, particularly for observing live cells in their native state. 6. Multiple choice questions MCQs. What is the primary advantage of phase contrast microscopy? A. High magnification power. B. Ability to observe live cells without staining. C. Three-dimensional imaging of specimens. D. Enhanced color contrast in specimens. Answer. B. Ability to observe live cells without staining. 2. Which component of a phase contrast microscope is responsible for creating phase shifts in light? A. Condenser lens. B. Phase plate. C. Objective lens. D. Eyepiece. Answer. B. Phase plate. A. What is a major limitation of phase contrast microscopy? Acquire staining for contrast. B. Only works for thick specimens. C. Produces halo artifacts around specimens. D. Cannot be used for live cell imaging. Answer. C. Produces halo artifacts around specimens. Phase contrast microscopy remains an essential technique in zoology, particularly for studying dynamic cellular processes without disrupting biological integrity. You said. Fluorescent and confocal microscopy. ChatGPT said. Fluorescent and confocal microscopy. Fluorescent and confocal microscopy are advanced optical techniques widely used in zoology and life sciences for visualizing specific biomolecules, organelles, and cellular processes with high precision. These methods utilize fluorescence to achieve enhanced contrast and resolution, allowing detailed examination of both fixed and live specimens. 1. Fluorescence microscopy. Principle of fluorescence microscopy. Fluorescence microscopy relies on the phenomenon of fluorescence, where certain molecules fluorophores absorb light at a specific excitation wavelength and emit light at a longer emission wavelength. This allows selective visualization of cellular components tagged with fluorescent dyes or proteins. A light source, e.g., mercury, xenon lamp, or LED, emits excitation light of a specific wavelength. A dichroic mirror reflects excitation light toward the sample but allows emitted fluorescence to pass through to the detector. A fluorescent dye, e.g., DAPI for DNA, FITC for proteins, binds to specific cell structures and fluoresces upon excitation. The emission filter ensures only fluorescence from a sample reaches the detector, improving contrast. Types of fluorescence microscopy. Epifluorescence microscopy. The most common fluorescence microscopy technique, where the entire specimen is illuminated and emitted light is detected. Suitable for fixed cells and some live cell imaging. B. Total internal reflection fluorescence (TIRF) microscopy. Selectively illuminates a thin section of the sample near the cover slip, approximately 100 to 200 nm, minimizing background noise. Ideal for studying membrane-associated events like receptor-ligand interactions. C. Super resolution microscopy. Light storm, stochastic optical reconstruction microscopy, and POM photoactivated localization microscopy surpass the diffraction limit, achieving nanometer scale resolution. Used for high-precision molecular and cellular studies. Applications of fluorescence microscopy in zoology. Localization of proteins. Fluorescently tagged antibodies help visualize protein distribution in cells. Live cell imaging. GFP, green fluorescent protein, allows real-time tracking of cellular events. Pathogen identification, used in microbiology to detect bacteria, viruses, and parasites. Neurobiology, helps study neural networks, synapses, and brain activity in live tissues. Limitations of fluorescence microscopy. Photobleaching, continuous exposure to excitation light can degrade fluorophores. Phototoxicity, high-intensity light may damage live cells. Out-of-focus fluorescence, blurred background fluorescence reduces image clarity, limiting resolution. 2. Confocal microscopy. Principle of confocal microscopy. Confocal microscopy overcomes the limitations of fluorescence microscopy by using a laser scanning system and pinhole optics to block out-of-focus light, resulting in sharp, high-contrast images. It enables optical sectioning, allowing 3D reconstruction of specimens. A laser source excites fluorophores in a focus plane. A pinhole aperture removes out-of-focus fluorescence, improving resolution. A scanning system, galvanometer mirrors, moves the laser across the specimen, capturing data point by point. A detector, PMT or CCD camera, records emitted fluorescence, constructing a high-resolution image. Types of confocal microscopy. A laser scanning confocal microscopy, LSCM, uses a single laser beam to scan the sample pixel by pixel. Produces detailed, high-resolution images but is slower than spinning disk confocal microscopy. B. Spinning disk confocal microscopy. Uses multiple pinholes on a rotating disk, allowing simultaneous imaging of multiple sample regions. Faster imaging with reduced phototoxicity, suitable for live cell studies. C. 2-photon microscopy. Uses 2 low-energy photons for excitation, reducing photobleaching and allowing deeper tissue penetration. Ideal for imaging thick biological samples, such as brain slices or embryos. Applications of confocal microscopy in zoology. Cellular and subcellular imaging, visualizing organelles, cytoskeletal structures, and proteins. Developmental biology, studying embryonic development in model organisms like zebrafish. Neuroscience, mapping brain activity and neuronal connectivity in real time. Parasitology and microbiology. You said, continue ChatGPT said. 2. Confocal microscopy, continued applications of confocal microscopy in zoology. Cellular and subcellular imaging. Confocal microscopy provides high-resolution imaging of organelles, cytoskeletal structures, and protein localization in cells. This is essential for studying cell division, intracellular transport, and signaling pathways. Developmental biology, used to study embryonic development in model organisms like Drosophila, zebrafish, and frogs by tracking gene expression and cell differentiation. Neuroscience. Confocal microscopy helps map brain activity, visualize neuronal networks, and study synaptic plasticity, contributing to research on neurodegenerative diseases. Parasitology and microbiology, used to analyze host pathogen interactions, bacterial biofilms, and the structure of parasites such as Plasmodium, malaria parasite, and Leishmania. Histopathology and disease research, helps in diagnosing diseases by imaging tissue samples with specific fluorescent markers. Advantages of confocal microscopy. Improved resolution and contrast. The pinhole aperture eliminates out-of-focus light, resulting in sharp images. 3D reconstruction. Optical sectioning allows scientists to create 3D models of biological specimens. Live cell imaging. Reduces phototoxicity when using optimized conditions, making it suitable for observing dynamic cellular processes. Deep tissue imaging. 2-photon confocal microscopy allows imaging of thick tissues with minimal damage. Limitations of confocal microscopy. Expensive and complex setup. Requires high-cost lasers, pinhole systems, and specialized detectors. Slower image acquisition. Laser scanning takes time, making it less suitable for extremely fast biological processes. Photoleaching and phototoxicity. Continuous laser exposure can degrade fluorophores and damage live cells, limiting long-term observations. Comparison of fluorescence and confocal microscopy. Feature fluorescence microscopy, confocal microscopy. Principal use is fluorophores to emit light after excitation. Uses lasers and pinhole optics for high-resolution imaging. Image quality lower resolution with background fluorescence. High resolution with optical sectioning. Depth penetration limited depth due to out-of-focus light. Deeper penetration, especially in 2-photon confocal microscopy. Live cell imaging suitable but can suffer from photobleaching. Better for live imaging with optimized settings. 3D reconstruction not possible. Possible due to optical sectioning. Speed faster but lower. Clarity slower but sharper. Images cost more. Affordable, expensive, and requires expertise. Solved previous year question. Describe the principle of confocal microscopy and discuss its advantages over fluorescence microscopy. Answer. Confocal microscopy enhances fluorescence imaging by using a pinhole aperture to remove out-of-focus light, resulting in high-resolution images. A laser excites fluorophores in a focused plane, and emitted light is detected through a photomultiplier tube, PMT, or CCD camera. Optical sectioning allows for 3D reconstruction of specimens. Advantages of confocal microscopy over fluorescence microscopy. Higher resolution, eliminates background fluorescence, producing clearer images. Optical sectioning, allows detailed 3D imaging of specimens. Reduced photobleaching, laser scanning minimizes prolonged exposure of the entire sample. Deeper tissue penetration, 2-photon confocal microscopy enables imaging of thick tissues. Thus, confocal microscopy is superior for studying intricate cellular structures and live biological processes. Multiple choice questions, MCQs. 1. What is the main advantage of confocal microscopy over fluorescence microscopy? A. Uses a stronger light source. B. Used without fluorophore. C. Produces high-resolution images by eliminating out-of-focus light. D. Uses a wider field of view. Answer. C. Produces high-resolution images by eliminating out-of-focus light. 2. Which component in confocal microscopy is responsible for removing out-of-focus fluorescence? A. Dichroic mirror. B. Pinhole aperture. C. Objective lens. D. Emission filter. Answer. B. Pinhole aperture. 3. Which type of confocal microscopy allows deeper tissue penetration and reduces photo damage? A. Spinning disc confocal microscopy. B. Epifluorescence microscopy. C. 2-photon confocal microscopy. D. Turf microscopy. Answer. C. 2-photon confocal microscopy. Conclusion. Fluorescence and confocal microscopy are essential tools in zoology for studying cells, tissues, and biomolecules with high specificity. While microscopy is widely used for its simplicity and fast imaging, confocal microscopy provides superior resolution, contrast, and 3D imaging capabilities, making it indispensable for advanced biological research. You said, electron microscopy. ChatGPT said, electron microscopy. Electron microscopy (EM) is a high-resolution imaging technique that uses a beam of electrons instead of light to visualize biological and non-biological specimens. It provides much higher magnification and resolution than optical microscopy, enabling the study of fine structural details of cells, organelles, viruses, and macromolecules. 1. Principle of electron microscopy. Microscopes operate on the principle that electrons have much shorter wavelengths than visible light, about 0.005 nanometers versus 400-700 nanometers. This allows them to resolve structures at the nanometer and even atomic scale. In electron microscopy, a high-voltage electron beam is generated using an electron gun, usually a tungsten filament or field emission source. The beam is focused by electromagnetic lenses instead of glass lenses as in light microscopes. The sample is placed in a vacuum chamber to prevent electron scattering by air molecules. The interaction between the electron beam and the specimen generates signals, such as transmitted electrons, scattered electrons, or secondary electrons, which are detected to form an image. 2. Types of electron microscopes. A. Transmission electron microscopy (TEM) principle. In TEM, a high-energy electron beam passes through an ultra-thin specimen, typically an LT, 100 nanometers thick. Some electrons are absorbed, scattered, or transmitted depending on the density of the specimen. The transmitted electrons are collected on a fluorescent screen or digital detector to create a highly detailed, two-dimensional 2D image. Applications of TEM. Structural analysis, study of cellular organelles like mitochondria, endoplasmic reticulum, ribosomes, and nuclear structures. Virus and bacteria imaging, used in virology and microbiology to study the morphology of viruses and bacteria. Macromolecular biology, helps determine the fine structure of proteins, DNA, and complexes using techniques like cryo-electron microscopy. Pathology and histology, used to study tissue morphology at the nanoscale. Advantages of TEM. Highest resolution among microscopy techniques, down to 0.1 nanometers. Detailed internal structure visualization of cells and macromolecules. Can be combined with cryo-electron microscopy for imaging biological samples in near-native states. Limitations of TEM. Extensive sample preparation, requires thin sectioning and staining with heavy metals, e.g., osmium tetroxide, uranyl acetate. Sample damage, high-energy electrons can degrade biological specimens. Expensive and requires expertise, operation and maintenance are costly. Beat scanning electron microscopy (SEM) principle. In SEM, a focused electron beam is scanned across the surface of the specimen. Electrons interact with the sample, generating secondary electrons, backscattered electrons, and X-rays. These signals are detected to create a 3D surface image of the sample. Applications of SEM. Surface morphology studies, used to examine the detailed topography of biological and non-biological samples. Entomology and zoology, used for studying insect exoskeletons, plant surfaces, and animal tissues. Nanotechnology and material science, helps visualize nanoparticles, fibers, and other nanomaterials. Forensic science, used in forensic investigations to analyze gunshot residues, fibers, and biological tissues. Advantages of SEM. High depth of field, provides a clear 3D image of a specimen surface. Minimal sample preparation compared to TEM, no ultra-thin sectioning required. Analyze large specimens, suitable for whole organisms, tissues, or materials. Limitations of SEM. Lower resolution than TEM, typically around 1 to 10 nm. Requires sample coating. Biological samples need to be coated with gold or platinum to prevent charging effects. Operates under vacuum, cannot be used for live cell imaging. 3. Comparison of TEM and SEM. Featured TEM SEM. Type 2D internal structure 3D surface morphology resolution higher, approximately 0.1 nanometers lower, approximately 1 to 10 nm, sample thickness ultra-thin sections, and LT, 100 nanometers intact specimens bulk samples, sample preparation complex, involves staining and sectioning simpler, involves coating with metal best for organelles, viruses, protein surface structures, topography costs very high but lower than TEM 4. Advanced electron microscopy techniques at cryo-electron microscopy, cryo-EM. Use frozen hydrated specimens to observe biological structures in a near-native state. Used for determining protein structures at atomic resolution. Beat environmental scanning electron microscopy, ESEM, allows imaging of hydrated and uncoated specimens by operating under a partial vacuum. Useful for studying biological samples in more natural conditions. C. Energy-directed X-ray spectroscopy, EDS, EDX, in SEM allows elemental composition analysis of a sample by detecting X-rays emitted during electron interactions. Used in material science and forensic applications. 5. Solved previous year question. Compare the principles and applications of TEM and SEM in biological research. Answer. Transmission electron microscopy, TEM, and scanning electron microscopy, SEM, are two major types of electron microscopy. Principle of TEM. A high-energy electron beam passes through an ultra-thin specimen. Transmitted electrons form a high-resolution 2D image of internal structures. Principle of SEM. A focused electron beam scans the surface of a specimen. Secondary and backscattered electrons create a 3D image of surface morphology. Applications of TEM. Imaging cell organelles, mitochondria, ribosomes, nuclei. Structural analysis of viruses and bacteria. Studying macromolecules using cryo-EM. Applications of SEM. Surface topology of biological tissues, exoskeletons, and materials. Forensic analysis, e.g., gunshot residues. Nanotechnology research. Comparison. TEM has higher resolution, 0.1 nanometers, but requires thin sections. SEM is lower resolution, 1-10 nanometers, but provides a 3D view of surfaces. Thus, TEM is preferred for detailed internal structures, while SEM is better for surface morphology analysis. 6. Multiple choice questions, MCQs. 1. What is the main reason electron microscopes achieve higher resolution than light microscopes? A. They use a stronger light source. B. Electrons have shorter wavelengths than visible light. C. They can operate in a vacuum. D. They use higher magnification lenses. Answer. B. Electrons have shorter wavelengths than visible light. 2. What type of electron microscope provides a three-dimensional surface image? A. Transmission electron microscope, TEM. B. Scanning electron microscope, SEM. C. Bright field microscope. D. Fluorescence microscope. Answer. B. Scanning electron microscope, SEM. 3. Why do biological samples in SEM need to be coated with gold or platinum? A. To enhance contrast. B. To prevent the sample from breaking. C. To avoid electron charging effects. D. To absorb excess electrons. Answer. C. To avoid electron charging effects. Conclusion. Electron microscopy is an indispensable tool in biological research, providing unparalleled resolution for studying cellular structures, viruses, and materials. While TEM is best for high-resolution internal imaging, SEM excels in 3D surface visualization. Both techniques have significantly advanced fields such as zoology, microbiology, nanotechnology, and forensic science. You said, SEM. ChatGPT said. Scanning electron microscopy, SEM. Scanning electron microscopy, SEM, is a powerful imaging technique that provides detailed 3D images of the surface morphology of biological and non-biological specimens. Unlike transmission electron microscopy, TEM, which transmits electrons through an ultra-thin sample to observe internal structures, SEM scans the surface of bulk samples to produce high-resolution images with excellent depth of field. 1. Principle of SEM. A focused electron beam produced by an electron gun is directed onto the surface of a specimen. The primary electrons interact with atoms in the sample, generating secondary electrons, backscattered electrons, and X-rays. These signals are detected and processed to form a high-resolution 3D image of the specimen's surface. The entire process occurs in a vacuum to prevent electron scattering by air molecules. 2. Components of an SEM. A. Electron source, electron gun. Generates a beam of electrons using a tungsten filament, lanthanum hexaboride source, or field emission gun, FEG. Field emission guns provide the highest resolution due to their smaller beam diameter. B. Electromagnetic lenses. Condenser lenses control the size and intensity of the electron beam. Objective lenses focus the beam onto the specimen. C. Scanning system. Electron beam is deflected in a raster pattern over the sample surface using scanning coils. The emitted signals are detected and converted into an image. D. Detectors. Secondary electron detector, SED, captures low-energy secondary electrons emitted from a sample surface, producing high-contrast images with fine details. Backscatter electron detector, BSD, detects high-energy electrons reflected from a sample, useful for differentiating materials based on atomic number contrast. X-ray detector, EDS, EDX, analyzes the elemental composition of a specimen by detecting characteristic X-rays. 3. Sample preparation in SEM. A. Fixation. Biological specimens are fixed using glutaraldehyde or osmium tetroxide to preserve structures. B. Dehydration. Water is removed using an ethanol or acetone gradient to prevent structural collapse. C. Drying. Critical point drying, CPD, is used to prevent surface distortion. D. Metal coating. A thin layer of gold, platinum, or palladium is sputter coated onto the sample to enhance conductivity and prevent charging. 4. Applications of SEM in zoology. A. Morphological studies. Used to study surface structures of biological samples, such as insect exoskeletons, plant trichomes, fish scales, and feathers. B. Taxonomy and systematics. SEM provides detailed images for species identification in insects, arthropods, and microorganisms. C. Parasitology. Used to examine external morphology of parasites like Plasmodium, malaria, leishmania, and trypanosoma. D. Paleontology and fossil studies. SEM helps analyze microfossils, ancient pollen grains, and fossilized tissues at high magnification. E. Material science and nanotechnology. Used for analyzing biomaterials, nanofibers, and biomedical implants. F. Forensic science. SEM is used for analyzing gunshot residues, hair fibers, and biological samples in forensic investigations. 5. Advantages of SEM. High resolution, can resolve surface structures up to 1 to 10 nm. 3D imaging, provides detailed surface morphology with high depth of field. Wide range of magnification, can magnify from 10x to 500,000x. Elemental analysis, energy dispersive X-ray spectroscopy EDS/EDX allows identification of elements in sample. 6. Limitations of SEM. Expensive equipment and maintenance, requires specialized facilities and trained operators. Complex sample preparation, biological samples need fixation, dehydration, and coating. Cannot image living cells, the vacuum environment and electron beam damage biological specimens. Limited internal structure imaging, unlike TEM, SEM cannot visualize internal cellular structures. 7. Comparison of SEM and TEM. Feature SEM TEM. Image type 3D surface morphology 2D internal structure resolution 1 to 10 nm 0.1 nanometers. Sample thickness no sectioning required ultra thin sections and LT 100 nanometers. Sample preparation coating with metal staining with heavy metals best for surface structures organelles, viruses, proteins cost lower than TEM higher. 8. Solved previous year question. Describe the working principle and applications of SEM in biological research. Answer. Scanning electron microscopy SEM is a high-resolution imaging technique used to examine surface structures of biological and non-biological specimens. Principle of SEM. A focused electron beam scans the specimen's surface in a raster pattern. The beam interacts with the sample, generating secondary electrons, backscattered electrons, and X-rays. These signals are collected by detectors to create a detailed 3D image of the surface morphology. Applications of SEM in biology. Morphological studies, used for analyzing the external features of insects, plant structures, and animal tissues. Taxonomy and identification, helps in species classification based on minute surface details. Forensic science, SEM is used in crime investigations to examine biological evidence like hair and fibers. Nanotechnology, used for studying nanoparticles, biomaterials, and drug delivery systems. Advantages. High depth of field provides excellent 3D visualization. Elemental analysis using energy dispersive X-ray spectroscopy EDS. Limitations. Sample preparation is complex, requiring metal coating. Biological samples must be dehydrated, making live cell imaging impossible. Thus, SEM is an essential tool in biological and forensic sciences, providing detailed surface morphology with high precision. 9. Multiple choice questions. MC1. What primary advantage of SEM over light microscopy? A. Higher depth of field and surface resolution. B. Ability to image live cells. C. Does not require sample preparation. D. Uses visible light instead of electrons. Answer. A. Higher depth of field and surface resolution. 2. Why biological samples in SEM require a metal coating? A. To make the sample conductive. B. To increase magnification. C. To preserve the sample in a vacuum. D. To absorb excess electrons. Answer. A. To make the sample conductive. 3. Which detector in SEM is primarily used to capture detailed surface images? A. X-ray detector. B. Secondary electron detector. C. Transmission detector. D. Fluorescence detector. Answer. B. Secondary electron detector. SED. And conclusion. Scanning electron microscopy SEM is a versatile tool in zoology, forensic science, and material research. It provides high-resolution 3D images of specimen surfaces, making it invaluable for studying biological structures, parasites, fossils, and nanomaterials. While SEM cannot image internal cell structures like TEM, its ability to visualize fine surface details has made it a critical technique in modern biological research. You said, TEM. ChatGPT said. Transmission electron microscopy TEM. Transmission electron microscopy TEM is an advanced imaging technique that provides high-resolution, two-dimensional 2D images of ultra-thin biological and non-biological specimens. Unlike scanning electron microscopy SEM, which provides surface details, TEM allows the visualization of internal structures at the molecular level. It is a crucial tool in cell biology, virology, nanotechnology, and material science. 1. Principle of TEM. A beam of high-energy electrons, accelerated up to 100 to 300 kV, is transmitted through an ultra-thin specimen, typically an LT, 100 nanometers. Some electrons pass through, while others are scattered or absorbed by the specimen. Transmitted electrons interact with a fluorescent screen or camera detector to form a high-resolution image. Areas where electrons pass through appear bright, while denser regions that scatter electrons appear dark. 2. Components of a TEM. Electron gun. Produces a beam of high-energy electrons using tungsten filament common lanthanum hexaboride lab for higher brightness field emission gun FEG for ultra-high resolution B electromagnetic lenses. Condenser lenses focus and control the intensity of the electron beam. Objective lenses provide the primary magnification. Projector lenses further magnify the image onto the detector. C. Sample holder. Holds the thin section specimen, usually placed on a copper grid coated with a support film. D. Image formation system. Fluorescent screen converts transmitted electrons into a visible image. CCD camera or direct electron detector captures digital images for analysis. 3. Sample preparation for TEM. Since TEM requires ultra-thin samples, extensive preparation is needed. A. Fixation. Biological samples are fixed with glutaraldehyde and osmium tetroxide to preserve ultrastructure. B. Dehydration. Samples are dehydrated using an ethanol or acetone gradient to remove water. C. Embedding. Specimens are embedded in epoxy or acrylic resin to provide mechanical support. D. Ultra-thin sectioning. Using an ultramicrodome, sections as thin as 50 to 100 nm are cut with a diamond or glass knife. E. Staining. Sections are stained with heavy metals e.g., uranium, lead, osmium to enhance contrast. F. Mounting on grids. Sections are placed on copper or nickel grids for examination. 4. Applications of TEM in zoology at cellular and molecular biology. Subcellular organelles. TEM allows detailed imaging of nuclei, mitochondria, ribosomes, Golgi bodies, and cytoskeleton. Viruses and bacteria. TEM is essential in virology for visualizing virus particles e.g., HIV, SARS-CoV-2, bacteriophages. B. Structural biology. Helps in studying proteins, nucleic acids, and macromolecules at atomic resolution. C. Histopathology and medical research. Used in cancer research and disease diagnosis to study abnormal cell structures. D. Nanotechnology and material science. Is used to analyze nanoparticles, biomaterials, and synthetic polymers. E. Forensic investigations. Helps in forensic research by analyzing tissue samples, fibers, and pathogens. 5. Advantages of TEM. Ultra-high resolution, can resolve structures up to 0.1 nanometers, revealing molecular details. Detailed internal structure imaging. Unlike SEM, which shows surface morphology, TEM provides internal structural details. Contrast enhancement with stains. Heavy metal staining improves visualization of cellular components. Useful for a wide range of biological and material studies. 6. Limitations of TEM. Extensive sample preparation, requires fixation, dehydration, and ultra-thin sectioning. High cost and maintenance. TEM instruments are expensive and require vacuum conditions. Cannot observe living cells. The vacuum and electron beam damage biological specimens. Complex interpretation, requires expertise in electron microscopy for image analysis. 7. Comparison of TEM and SEM. Feature TEM SEM. Image type 2D internal structure 3D surface morphology resolution 0.1 nanometers 1 to 10 nm. Sample thickness in LT, 100 nanometers ultra-thin sections, no sectioning needed sample preparation extensive fixation, embedding, sectioning coating with metal best for organelles, proteins, viruses surface structures 8. Solved previous year question. Explain the working principle, sample preparation, and applications of transmission electron microscopy TEM in biological sciences. Answer. Principle of TEM. A high-energy electron beam, 100-300 kV, is transmitted through an ultra-thin specimen. Some electrons pass through, while others are scattered or absorbed by the sample. The transmitted electrons interact with a fluorescent screen or CCD detector to form a high-resolution image. Sample preparation in TEM. Fixation. Glutaraldehyde and osmium tetroxide are used to preserve cellular structures. Dehydration. The sample is dehydrated with ethanol or acetone. Embedding. The specimen is embedded in epoxy resin for mechanical support. Sectioning. Thin sections 50-100 nanometers are cut using an ultramicrodome. Staining. Heavy metals e.g., uranium, lead enhance contrast. Mounting. The sample is placed on a copper grid for imaging. Applications of TEM in biology. Cell biology. Studying organelles, mitochondria, ribosomes, Golgi bodies. Virology. Imaging viruses, HIV, SARS-CoV-2. Histopathology. Detecting cancer and disease cells. Nanotechnology. Analyzing nanoparticles and biomaterials. Advantages. High-resolution imaging of internal structures. Useful for studying molecular and cellular ultrastructure. Limitations. Requires complex sample preparation. Cannot image live cells due to vacuum conditions. TEM is a powerful tool in structural biology, pathology, and nanotechnology. 9. Multiple choice questions MCQs 1. What is the primary advantage of TEM over light microscopy? A. Ability to image live cells. B. Higher resolution and visualization of internal structure. C. Requires no sample preparation. D. Uses visible light instead of electrons. Answer. B. Higher resolution and visualization of internal structures. 2. Why are biological samples stained with heavy metals in TEM? A. To make the sample conductive. B. To increase electron scattering and enhance contrast. C. To dehydrate the specimen. D. To absorb excess electrons. Answer. B. To increase electron scattering and enhance contrast. 3. What is the function of the objective lens in TEM? A. Produces the primary magnification. B. Generates the electron beam. C. Collects backscattered electrons. D. Converts electrons into light signals. Answer. A. Produces the primary magnification. 10. Conclusion. Transmission electron microscopy TEM is a critical tool for high-resolution imaging of biological and material samples. It allows visualization of subcellular structures, viruses, and nanomaterials with atomic-level precision. Though it requires extensive sample preparation and operates in a vacuum, its unmatched resolution makes it indispensable in cell biology, structural biology, virology, and nanotechnology. Freeze etch. ChatGPT said, freeze etching microscopy. Freezing is a specialized electron microscopy (EM) technique used to study biological membranes, cellular organelles, and macromolecular structures in their near-native state. This technique involves rapidly freezing a sample, followed by controlled fracturing and etching to expose internal structures, and then imaging using transmission electron microscopy (TEM) or scanning electron microscopy (SEM). 1. Principle of freeze etching. The freeze etching technique is based on 3 main steps. Rapid freezing. The biological sample is quickly frozen in liquid nitrogen or helium, preventing the formation of large ice crystals that can distort structures. Fracturing. The frozen sample is physically fractured, usually along natural planes like membranes, exposing internal components. Etching. The fractured surface is partially warmed under vacuum to sublimate a thin layer of ice, enhancing contrast. Replicating and imaging. A thin metal carbon replica is created and observed under temp. This method allows direct visualization of cellular membranes, organelles, and protein complexes with minimal artifacts. 2. Steps in freeze etching at sample preparation. The specimen, e.g., a cell, tissue, or macromolecule, is suspended in a buffer to maintain its native environment. It is rapidly frozen in liquid nitrogen -196 degrees Celsius or liquid helium -269 degrees Celsius using ultra-fast plunging techniques. B. Freeze fracture. The frozen specimen is transferred to a vacuum chamber and fractured using a microblade or sharp knife. The fracture occurs along weak molecular interfaces, typically membranes and lipid bilayers, revealing transmembrane structures. C. Etching, sublimation. The sample is warmed slightly, about -100 degrees Celsius, under high vacuum, allowing sublimation of surface ice. This enhances the contrast of surface structures by removing unwanted frozen water layers. D. Replication and imaging. A thin film of platinum or carbon is deposited onto the exposed structures to create a replica. The underlying biological material is dissolved, leaving behind a detailed replica of the fractured surface. The replica is then analyzed under temp for high-resolution imaging. 3. Applications of freeze etching in zoology at membrane biology. Freeze etching is widely used to study cell membranes, lipid bilayers, and membrane proteins. It reveals intramembranous particles, which correspond to transmembrane proteins. B. Cell and organelle structure. Allows visualization of organelles like mitochondria, nuclei, Golgi apparatus, and endoplasmic reticulum. C. Virus and bacteria studies. Provides insights into viral capsids, bacterial cell walls, and flagella without distortion. D. Neuroscience. Used for imaging synapses, neuronal membranes, and ion channels. E. Biofilms and microbial communities. Helps study the organization of bacterial biofilms and microbial interactions. 4. Advantages of freeze etching. Native structures. Unlike chemical fixation, freeze etching retains biological molecules in their natural state. Minimizes artifacts. Avoids artifacts caused by dehydration and fixation in traditional TEM methods. Reveals internal membrane structures. Provides direct insights into membrane architecture and protein organization. High-resolution imaging. Produces nanometer-scale details, making it useful for cellular and molecular studies. 5. Limitations of freeze etching. Complex sample preparation. Requires specialized equipment and expertise. Requires a vacuum environment. The sample must be handled in a high vacuum chamber, making in vivo studies impossible. Art fracturing. Some structural components may not fracture cleanly, leading to interpretation challenges. 6. Comparison of freeze etching and traditional TEM. Feature freeze etching TEM conventional TEM. Rapid freezing, fracture, and etching chemical fixation, dehydration, embedding structural preservation excellent moderate, fixation artifacts membrane analysis detailed, reveals transmembrane proteins limited contrast artifacts few or more due to chemical treatment 7. Solved previous year question. Explain the principle, steps, and applications of the freeze etching technique in electron microscopy. Answer. Principle. Freeze etching is an advanced electron microscopy technique that involves rapid freezing, fracturing, and sublimation to expose cellular structures in their native state. The fractured surface is coated with a metal carbon replica and imaged using transmission electron microscopy TEM. Steps. Rapid freezing. The specimen is frozen at -196 degrees Celsius to preserve structures. Fracturing. The frozen sample is fractured, exposing internal membranes and organelles. Etching. A thin layer of ice is sublimated to enhance contrast. Replica formation. A thin platinum carbon coating is applied to create a durable replica. Imaging. The replica is examined under TEM for structural details. Applications. Membrane biology. Studies lipid bilayers and transmembrane proteins. Cell biology. Analyzes organelles like mitochondria and Golgi bodies. Microbiology. Examines bacteria, viruses, and microbial biofilms. Neuroscience. Investigates synaptic vesicles and ion channels. Advantages. Preserves native structures without artifacts. Provides high-resolution images of cellular membranes. Limitations. Requires specialized equipment and expertise. Not suitable for live cell imaging due to vacuum conditions. Thus, freeze etching is a powerful tool for biological imaging, offering insights into membrane architecture, protein organization, and ultrastructural details. 8. Multiple choice questions MCQs 1. What is the main advantage of freeze etching over conventional TEM? A. It provides 3D images. B. It avoids chemical fixation artifacts. C. It allows live cell imaging. D. It uses visible light instead of electrons. Answer. B. It avoids chemical fixation artifacts. 2. What is the purpose of the etching step in freeze etching? A. To coat the sample with a metal replica. B. To remove a thin layer of ice and enhance contrast. C. To stain the sample with heavy metals. D. To make the sample conductive for SEM. Answer. B. To remove a thin layer of ice and enhance contrast. 3. In freeze etching, what is the main component used for making replicas? A. Silver. B. Gold. C. Carbon and platinum. D. Aluminum. Answer. C. Carbon and platinum. 9. Conclusion. Freeze etching is an advanced electron microscopy technique that allows high-resolution imaging of membranes, organelles, and macromolecules while preserving their native structure. This method is particularly valuable in cell biology, microbiology, and neuroscience, where detailed ultrastructural analysis is required. Despite its complex preparation, the benefits of artifact-free imaging make it an essential tool in modern biological research. You said. Freeze fracture methods. ChatGPT said. Freeze fracture methods. Freeze fracture is a specialized electron microscopy technique used to study the ultrastructure of biological membranes by splitting frozen specimens along their natural planes. It provides unique insights into membrane organization, lipid bilayers, and transmembrane proteins. This method is often used in cell biology, microbiology, and neuroscience to visualize membrane-bound structures at a nanometer scale. 1. Principle of freeze fracture. Freeze fracture microscopy involves freezing a biological sample at ultra-low temperatures and then physically fracturing it to reveal internal membrane structures. The technique exploits the fact that frozen biological specimens tend to break along lines of weakness, which are usually lipid bilayers of membranes. The fractured surface is coated with a thin layer of platinum and carbon, forming a replica. The biological material is then dissolved, leaving behind an electron-dense replica that can be examined using transmission electron microscopy (TEM). This method allows the visualization of integral membrane proteins, lipid domains, and cellular compartments with minimal distortion. 2. Steps in freeze fracture technique at sample preparation. The biological specimen, e.g., cells, tissues, organelles, is suspended in a cryoprotectant solution to prevent ice crystal formation. The sample is rapidly frozen in liquid nitrogen -196 degrees Celsius or liquid helium -269 degrees Celsius to preserve its natural state. B. Freeze fracturing. The frozen specimen is placed in a vacuum chamber and fractured using a microblade, diamond knife, or metal rod. The fracture typically follows membrane lipid bilayers, revealing intramembrane structures such as proteins, ion channels, and lipid rafts. The two fractured membrane faces are protoplasmic face P face, the inner leaflet of the lipid bilayer, which contains most transmembrane proteins. Exoplasmic face E face, the outer leaflet of the bilayer, usually showing fewer protein structures. C. Replication. A thin platinum carbon layer is deposited onto the fractured surface to form a replica of the exposed structures. The biological material is then dissolved, leaving the metal carbon replica intact. D. Imaging under TEM. The replica is placed on an EM grid and examined under transmission electron microscopy (TEM). This provides high-resolution images of membrane structures, proteins, and lipid domains. 3. Applications of freeze fracture. A. Membrane structure analysis. Provides direct visualization of lipid bilayers and their associated proteins. Used to study membrane fluidity, lipid domains, and cholesterol distribution. B. Transmembrane protein organization. Reveals the distribution and density of integral membrane proteins, ion channels, and receptors. Used in neuroscience to study synaptic vesicles and neurotransmitter receptors. C. Cell junctions and adhesion structures. To examine tight junctions, desmosomes, and gap junctions in epithelial and neural tissues. Helps understand cell-cell communication and adhesion mechanisms. D. Bacterial and viral studies. Reveals bacterial outer membranes, flagella, and fimbriae. Used to study the structure of viral envelopes and capsids. E. Drug effects on membranes. Used in pharmacology to analyze how drugs and toxins affect membrane integrity. Helps in studying membrane permeability changes induced by antibiotics, detergents, or cholesterol-altering compounds. 4. Advantages of freeze fracture. Preserves native membrane structures without chemical fixation artifacts. Provides high-resolution images of lipid bilayers and membrane proteins. Reveals transmembrane protein distribution and clustering. Avoids dehydration effects seen in conventional electron microscopy. 5. Limitations of freeze fracture. Requires specialized equipment and expertise. Fracture planes may not always be predictable, leading to inconsistencies. Sample handling is complex, requiring ultra-low temperatures and vacuum conditions. Only provides static images, making it unsuitable for dynamic studies of live cells. 6. Comparison of freeze fracture and freeze etching feature. Freeze fracture, freeze etching. Purpose review. Membrane structure and transmembrane proteins enhances contrast by sublimating surface ice steps. Freezing, right pointing arrow. Fracturing, right pointing arrow. Replication, right pointing arrow. TEM imaging. Freezing, right pointing arrow. Fracturing, right pointing arrow. Etching, right pointing arrow. Replication, right pointing arrow. TEM imaging. Main use: studying lipid bilayers, ion channels, and proteins. Studying detailed surface textures and subcellular structures. Image clarity. High contrast for membrane proteins. High contrast for exposed surfaces. 7. Solved previous year question. Question. Describe the freeze fracture technique and its applications in studying biological membranes. Answer. Introduction. Freeze fracture is an electron microscopy technique used to analyze biological membranes and their associated proteins by physically splitting frozen specimens. This method helps visualize membrane organization, protein distribution, and lipid domains with nanometer scale resolution. Principle. The specimen is rapidly frozen to preserve its native state. The sample is fractured along membrane lipid bilayers, revealing intramembrane proteins and membrane-associated structures. A platinum carbon replica is created and examined under transmission electron microscopy (TEM). Steps. Rapid freezing. The specimen is frozen in liquid nitrogen -196 degrees Celsius to prevent ice crystal formation. Fracturing. The frozen sample is fractured, exposing membrane structures. Replica formation. A metal carbon layer is deposited onto the exposed surface. Dissolution of biological material. The biological specimen is removed, leaving the replica intact. TEM imaging. The replica is analyzed under TEM to visualize membrane details. Applications. Membrane studies. Analyzes lipid bilayers, membrane proteins, and cholesterol distribution. Neuroscience. Studies synaptic vesicles, neurotransmitter receptors, and neuronal membranes. Microbiology. Examines bacterial outer membranes, viral envelopes, and flagella. Cell junctions. Reveals tight junctions, gap junctions, and desmosomes. Advantages. Provides high-resolution images of transmembrane protein. Preserves the native structure of membranes. Avoids chemical fixation artifacts. Limitations. Requires specialized cryo equipment and ultra-low temperatures. Fracture planes may be unpredictable, affecting image consistency. Thus, freeze fracture microscopy is a powerful tool for studying membrane structure and function, helping to understand cell biology, neuroscience, and microbiology at the ultrastructural level. 8. Multiple choice questions MCQs. 1. Which face of the fractured membrane contains more transmembrane proteins? A. E face, exoplasmic face. B face, protoplasmic face. C. Both contain equal amounts. D. None of the above. Answer. B. D face, protoplasmic face. 2. What is the main advantage of freeze fracture microscopy? A. Allows live cell imaging. B. Reveals transmembrane proteins and lipid bilayers. C. Uses visible light instead of electrons. D. Requires no vacuum environment. Answer. B. Reveals transmembrane proteins and lipid bilayers. 3. Which of the following is not a step in freeze fracture? A. Rapid freezing. B. Fracturing along lipid bilayers. C. Staining with fluorescent dyes. D. TEM imaging of metal carbon replicas. Answer. C. Staining with fluorescent dyes. 9. Conclusion. Freeze fracture is a powerful electron microscopy technique that enables direct visualization of membrane organization, transmembrane proteins, and lipid bilayers. It plays a crucial role in cell biology, microbiology, and neuroscience, providing high-resolution images with minimal artifacts. You said. Hey, can you stop giving tables please? Only paragraph. Okay, ChatGPT said. Memory updated. Got it. I'll continue with only paragraph-based explanations from now on. Let me know the next topic, and I'll provide a detailed lecture-style note for it. You said, cytophotometry. ChatGPT said, cytophotometry. Cytophotometry is a powerful analytical technique used to quantify chemical components within individual cells by measuring the absorption or fluorescence of specific cellular constituents. This method is particularly valuable in cell biology, genetics, and pathology, where it helps analyze DNA, RNA, proteins, and enzyme activities in different cellular compartments. Cytophotometry is based on the principles of photometry, where light intensity changes due to interaction with stained biological materials, allowing precise quantification of cellular components. Principle of cytophotometry. The core concept of cytophotometry is based on the Beer-Lambert law, which states that the amount of light absorbed by a substance is proportional to its concentration. In cytophotometry, biological samples are treated with specific stains that bind to particular cellular components. The stained samples are then analyzed using a photometer attached to a microscope, which measures the absorbance or fluorescence of light at specific wavelengths. The intensity of the signal is directly correlated with the quantity of the targeted cellular material, such as DNA, RNA, or proteins. Types of cytophotometry. There are two primary types of cytophotometry: absorption cytophotometry and fluorescence cytophotometry. Absorption cytophotometry. This method relies on the measurement of light absorbed by stained cellular components. Common stains include fuliginous stain for DNA and bromophenol blue for proteins. It is used to determine DNA ploidy, nuclear abnormalities, and enzyme activity in cells. Fluorescence cytophotometry. Uses fluorescent dyes that emit light upon excitation by a specific wavelength. Dyes like acridine orange and DAPI are commonly used to quantify nucleic acids. This method is sensitive in absorption cytophotometry and allows the detection of extremely small amounts of cellular components. Applications of cytophotometry. Cytophotometry has a wide range of applications in biological and medical sciences. DNA quantification and ploidy analysis. Used to measure DNA content in individual cells, helping in the identification of aneuploidy and polyploidy in cancer cells and plant cells. Fuliginous staining combined with cytophotometry provides highly accurate measurements of DNA concentration. Cell cycle analysis. Helps determine the proportion of cells in different phases of the cell cycle G1, S, G2, and M phases. Used in cancer research to study cell proliferation rates in response to chemotherapy. Enzyme histochemistry. Cytophotometry can measure enzymatic activity in tissues and cells by quantifying reaction products that absorb or fluoresce light. Commonly used to study oxidative enzymes, peroxidases, and phosphatases in tissues. Protein and RNA quantification. Specific stains like pyron and Y for RNA and bromophenol blue for proteins enable quantitative analysis of cellular components. Helps in studying cellular differentiation, gene expression, and protein synthesis. Pathology and cancer diagnosis. Used in clinical diagnostics to differentiate between benign and malignant cells based on DNA content. Helps in grading tumors and predicting their progression by analyzing nuclear abnormalities. Plant and microbial cytology. Applied in botany to study ploidy levels in plants and track genetic modifications. Used in microbiology to assess bacterial DNA content and cell viability. Advantages of cytophotometry. Cytophotometry provides precise and reproducible measurements of cellular components. It is a non-destructive technique, allowing researchers to analyze individual cells while preserving their structure. The method enables high sensitivity and specificity in detecting nucleic acids, proteins, and enzyme activities. Furthermore, it is useful for single-cell analysis, making it a critical tool in cancer research, cytogenetics, and developmental biology. Limitations of cytophotometry. Despite its advantages, cytophotometry has some limitations. It requires highly specific staining protocols, and any variation in staining can affect results. The technique is also limited to fixed cells, meaning it cannot be used for real-time live cell analysis. Additionally, it may be less effective in complex tissues where background staining can interfere with measurements. The accuracy of the results depends on the quality of sample preparation and the sensitivity of a cytophotometric system. Solved previous year question. Question: how is cytophotometry used in DNA quantification, and what are its applications in cancer research? Answer: cytophotometry is a technique used for DNA quantification by measuring light absorption or fluorescence from stained cellular components. The fuliginous reaction is commonly used for DNA staining, where Schiff's reagent binds specifically to deoxyribose in DNA, producing a magenta-colored complex that can be quantified using photometric methods. This allows for precise determination of DNA content in individual cells. In cancer research, cytophotometry plays a crucial role in tumor grading and prognosis. Cancer cells often exhibit aneuploidy, abnormal DNA content, which can be detected using cytophotometric analysis. The technique is used to measure DNA ploidy levels, helping differentiate between normal, pre-malignant, and malignant cells. By analyzing cell cycle distribution, cytophotometry provides insights into tumor aggressiveness in response to treatment. It is also used to evaluate chemotherapy effects by assessing changes in DNA synthesis and cell proliferation rates. Thus, cytophotometry is an essential tool for understanding cancer biology, genetic disorders, and cellular responses to therapeutic agents. You said, micrometry. ChatGPT said, micrometry. Micrometry is a technique used for the precise measurement of microscopic objects such as cells, organelles, and microorganisms. It involves the use of a micrometer scale, also called an eyepiece radical or stage micrometer, along with a microscope to determine the dimensions of structures that are too small to be measured with the naked eye. Micrometry is widely used in biology, medicine, materials science, and forensic science to analyze cellular morphology, tissue structures, and microscopic particles. Principle of micrometry. Micrometry is based on optical magnification and calibration. A special micrometer scale is placed in the eyepiece or on the microscope stage, and its divisions are used to determine the size of the specimen being observed. The method relies on the comparison between the known distance of a micrometer scale and the unknown size of the specimen, allowing accurate measurements at different magnifications. There are two types of micrometers commonly used in microscopy. Stick micrometer, a glass slide with a precisely marked scale, usually in micrometers, placed on the microscope.